So we use a tool called a pedigree to track the relationship of traits across multiple generations, so from parents to children through many generations. So this is what a pedigree looks like, and it's basically just a family tree. So a square is a male, and a circle is a female. And then the shading makes a difference. So each pedigree that we will look at is going to be concerned with some trait. So if a block is shaded in, that means that they have the trait, whatever it is that we're looking at. In this specific pedigree, the trait that we're looking at is a widow's peak. Usually pedigrees are looked at to measure diseases through family trees. This one is just looking at a specific trait, which is the widow's peak. In this case, a widow's peak is a dominant trait. So anybody who's shaded in purple is either going to be a male with a widow's peak or a female with a widow's peak. And the widow's peak is the little peak there on the hairline on the forehead. Anybody who is not shaded in, so these are going to be white. The shaded colors can be any color. They're usually just black or white. This means that they do not have the trait. So this person would have no widow's peak. The line between two partners is called a mating or marriage line. So that indicates that this male and this female mated and had some offspring. And then these are their offspring, and they're always in the order of birth. So this is the first daughter, and then this is the second daughter. There are different kinds of genetic disorders that we use pedigrees to examine. So we'll talk about some recessive alleles here. So recessively inherited disorders only show up in individuals who are homozygous recessive. So in this case, the example down here is albinism. And albinism is a single allele recessive disorder. So you'd have to be little a to be albino. And albino means that you don't have pigmentation in your hair and your skin. So you can see in the picture that the girl on the right is albino. She doesn't have any pigmentation in the hair, whereas her sister here is normal and does have pigmentation in the hair and the skin. Recessive alleles can be carried through generations in people called carriers. So somebody who is big A for normal pigmentation and little a would be normal, right? This is a dominant recessive genetic situation. So big A would cover up little a, and this person would look normal. However, if somebody who is big A little a mates with somebody else who's big A little a, they have a 1/4 chance or 25% chance of having a baby who is albino. So most people who have recessive disorders are born to parents who are carriers. And that's usually because most people who have recessive disorders, a lot of recessive disorders can be extremely serious, and so those people don't tend to reproduce. Albinism is one of the ones that does not affect reproduction, so as long as you take measures to protect yourself from the sun, an albino person doesn't necessarily have any worse chance at reproducing. So in a different recessive allele, one that causes a disease, if the disease is very rare, it's unlikely that two carriers will meet because the frequency of that recessive allele in the population is so low. And so usually the frequency of recessive alleles and recessive diseases is fairly low. Consanguineous meeting, so con means close blood matings. So this would be matings between very close family members. Those tend to increase the chance of carriers meeting with other carriers and carrying the same rare alleles. So in our society, we call this incest, and we have taboos and laws against marriage of close relatives. Most animal societies actually have mechanisms in place to prevent close mating like this so that rare genetic disorders don't meet each other in close families. So this is kind of evolved in society as a natural selection against rare genetic disorders. Cystic fibrosis is another lethal genetic disorder. So this is a very common one. So this would be 1 out of every 2,500 people of European, of European descent. What happens with cystic fibrosis is the linings of the inside of the lungs and the digestive system become very thick. They get a buildup of chloride outside of the cells, and they're not able to absorb or transfer nutrients across those cells. And so people with cystic fibrosis often have issues with breathing in their lungs. One way to treat this is a lung transplant. And then sometimes they have issues with absorbing nutrients in the small intestine. And so this can be lethal if not treated. Another homozygous recessive disorder is called sickle cell disease. So sickle cell disease is about 1 out of every 400 people of African American descent. And what happens is the red blood cells, instead of being circular like they normally should be, end up being sickle-shaped. And these sickle-shaped cells can tend to build up on each other and cause clotting. And so this can get stuck in joints and cause pain. They're also not as good at absorbing oxygen and carrying oxygen. So somebody who is homozygous, so little r, would have sickle cell disease, and all of their cells would be sickled like this. Somebody who's a heterozygous, so big R, little r, says they have what's called sickle cell trait. So about half of their cells would be normal, and then another half would be sickle. And so they're usually healthy, but they might suffer some symptoms of sickle cell disease, but they generally live and are able to reproduce. And so this is how this trait continues to be carried on in the population, because carriers are okay, and then they pass that trait on. So if somebody who's a carrier mates with somebody else who's a carrier, they again have a 25% chance of getting someone who has the full-on sickle cell disease. So you would imagine that something like this would get selected out of the population. Eventually, the people who have sickle cell trait would be less fit to mate and reproduce, and then eventually these kind of diseases should go away. However, this is fairly common in African Americans, and it's 1 out of 400, and it's fairly common in areas in Africa where malaria is common. So you can see these two maps. This map on the left shows all the pink where malaria is common, so kind of western to central Africa here. And then over here is a little heat map showing the allele frequency of the sickle cell. So HS is the sickle cell. And so it's common also in kind of the same area. These really overlap here in this central western Africa. And the reason for that is because even though having sickle cell disease is damaging, in areas where malaria is common, having sickle cell trait, the heterozygous, so HS is what they're using here, is actually an advantage. This is something called heterozygote advantage. What happens is those people are generally okay. They might have a few symptoms, but they're generally healthy. And because some of their cell is sickled instead of regular, it actually helps to prevent them against malaria. Malaria, when it goes through a life cycle, has to actually reproduce in red blood cells, and it can't get into the sickled red blood cells. And so this is one of the reasons why the frequency of this disease is still very high, because having sickle cell trait makes you less likely to die of malaria. So sickle cell disease is not very common in first world westernized countries like the United States, where we don't have to deal with things like malaria. However, in Western and Central Africa, where malaria is still a huge problem, the frequency of sickle cell disease is actually much higher because it confers some advantage against dying of malaria. Other disorders can be inherited with dominant alleles. These are generally rare, and they usually arise by some kind of mutation. So, for example, achondroplasia is a form of dwarfism. There's several different types of dwarfism. This is just one of them specifically. And if you have capital D, because this is a dominant trait, this would be dwarfed. And this is a dominant disorder, so even somebody who only has one capital D would still have the dwarf trait, and then the only person who would be of normal height would be a heterozygote recessive. So that's what we mean here by it's a dominant disorder. Anybody who has a dominant allele is going to show that disorder. Typically, if a dominant disorder is lethal, that individual is not going to be able to reproduce and pass that trait on. That's another reason why dominant disorders are rare. Dwarfism, like achondroplasia, doesn't affect the ability to reproduce, and so that trait gets passed on because it's not lethal. Huntington's disease is a neurodegenerative disease, which over time prevents the brain from communicating with the muscles and actually degenerates neurons, and it's lethal. However, Huntington's doesn't set until about 30 to 40 years of age. So most people, by the time they're 30 to 40 years of age, have had the window of opportunity to already reproduce and pass that gene on to their offspring. So let's imagine if an individual finds out that they have Huntington's disease. So they would be big H, little h. It's not very likely that they'd be homozygous dominant. Let's say that they mated with somebody who did not have Huntington's disease. So little h, little h. What is the chance that their offspring is going to also have Huntington's disease? So the chance that they have a baby, right, these are their offsprings, is actually 50% that they will have Huntington's. And only 50% that they'll be normal. Right, so little h is normal. Big H, little h means they'll have Huntington's. So dominant disorders have a high rate of transmission if they're able to be passed on. So it's a 50% chance. So somebody who finds out they have Huntington's would want to do genetic panels on any of their children to find out if they also have Huntington's. We do a lot of testing nowadays with modern medicine to look for genetic disorders so that parents are aware of their chances of passing on genetic traits, as well as prepared to deal with any genetic disorders that they find in infants. Most women who are pregnant will go through genetic screenings where they will actually look at the maternal blood because some fetal cells do actually get into the maternal blood, and they can look for chromosomal disorders that way. If those tend to show that there's likely a genetic disorder or somebody who's coming from a family that likely has genetic disorders, they might actually get more invasive testing. So amniocentesis is done a little earlier in the pregnancy, and this is where they would pull some of the liquid from the amniotic fluid, so that's the water that surrounds the fetus. And what happens is some of the fetal cells slough off, and they can do genetic testing on those to see if the baby has any genetic disorders. Chorionic villi sampling is actually taking a small piece of the placenta, and this again is going to have some fetal cells, and they can do the same kind of genetic testing. Both of those, while very informative, do carry a slight risk to miscarriage, to damage of the baby and the pregnancy. Most other techniques like ultrasound, maternal blood draw are usually just done visually in utero. When a baby is born, they take a ton of blood samples, and then they do tests for other genetic disorders at that point. And then there's other genetic disorders that are tested for throughout life as children age. Here I've got two examples of pedigrees, one of which is a dominant trait and one of which is a recessive. So I want to give you some practice here at recognizing dominant and recessive disorders. So the biggest thing to look for is carriers. A carrier will be somebody who does not show the trait, right? However, can pass it on to an offspring who does show the trait. So carriers are only going to occur in recessive genetic disorders. So if we look at the pedigree on the left, we see a male here who has the trait, who has passed it on to a daughter and a son. A female here who has this trait has passed it on to a daughter. And then these two both had the trait and they passed it on to one child. So I don't see any carriers in here. So a carrier would be somebody who does not have the trait, who passed it on to someone who does have the trait. Let's look over here. I see two people who do not, but they have two children who do have the trait. So these two individuals are going to be carriers. So that tells me that this is a recessive disorder. What you can do with a pedigree, once you've identified, either you'll have to identify that it's a recessive disorder, or I will just tell you in the test question. Now I can figure out the genotypes of these individuals. I know this is a recessive disorder. I'm just going to use A. So this person has to be big A, little a, big A, little a. I don't know this individual because they don't have the trait. So they're either big A, or big A, little a, but I'm not sure. These two individuals do have this trait, so I know it's a recessive disorder, which means if they have the trait, they are homozygous recessive. That's the only way to have a recessive disease. So these two individuals have to be little a, little a. I can actually go ahead and fill that in for all of the individuals that have that trait. This individual right here does not have the trait. However, they do have an offspring that does, which means they have to have at least one little a. The only way that this person has the trait is if they get one little a from dad and one little a from mom. So I know that her genetic genotype is big A, little a. Down here, we have two people that do not have the trait, so they have at least one big A. However, they have an offspring that has a little a, little a. So they each have to have a little a, carriers here, that have passed the trait on to an offspring. This one here we know has a big A, but we don't know the second one because we don't have any more information. So you'll likely see at least one or two multiple choice questions where I ask you about a specific individual and ask you either to give me their genotype on a pedigree or what their chance of passing on a specific genetic trait is to their offspring. So this is actually an example of a dominant disorder. There are some distinctive characteristics of dominant disorders that you can see. And so here, one is that if a child has it, the parent always has it. So here we can see a child with parents that have the trait, child with parents that have the trait, child with parents that have the trait. So a dominant disorder is going to show up in every generation. Whereas the recessives, you can see skip generations. This parent had it, it skipped this child, but it showed up again in this generation. So it's kind of skipping from here to here. Another characteristic of a dominant disorder is that about 1/2 of the children are affected. So just like we saw in the Huntington's pedigree or Huntington's Punnett square, there's a 50% chance of passing these traits on to the offspring. So here we've got 4 children and 2 out of 4 are affected. Here we've got 2 children, 1 out of 2 is affected. Here we've got 2 children, 1 out of 2 is affected. So this is not perfect, but it's a good way to kind of distinguish dominant from recessive. So here we know that this is a dominant disorder, which means that they could be either big A, or big A, little a to have the disorder. So what we can do is we can go to all the people who do not have the disorder and we can write in their genotypes. We know that all the people who do not have, we can say this is some generic dominant disorder, are going to be little a, because they do not show the disorder. So somebody who is normal would be little a. And then we can try to fill in some of these dominant individuals. So in order for this person to have gotten 2 little a's, they would have gotten 1 little a from dad here, and then they would have gotten 1 little a from mom. So this individual has to be big A, little a. Same thing for here because they have 2 offspring without it, so this person has to be big A, little a. These children also have to be big A, little a because they can only get little a's from one parent. So this person has the trait because they got the big A from dad, but mom can only give little a. So these have to also be heterozygous. Same thing over here. This girl got her big A trait from mom, but dad would only have been able to pass on a little a, so she has to be heterozygous. This individual we actually can't decide without more information because they could get big A from both parents, or they could have gotten a big A and a little a from either parent. So this one is big A, or big A, little a. We wouldn't be able to figure this one out without more information.